Presentation to what I think will be another exciting presentation, which is an update on AMX0035, which has a name now, Relyvrio. And Jamie Timmons is going to join us to talk about AMX0035. Jamie is the head of scientific communications at Amylyx and works closely with Josh Cohen and Justin Klee, who are co-founders and co-CEOs of Amylyx Pharmaceuticals as well.
Great, thank you so much. I'm just going to share our slides here.
Thanks for being here, Jamie.
Great. So on behalf of Amylyx, thank you to ALS ONE for the opportunity to speak today. Some of you may know I had the really important role of being our data presenter and the moderator for our two advisory committees. So it's really my pleasure to kind of take a step back today, maybe slightly less intense of an arena, to really go through our data and to share where we stand at the moment. It's been an exciting week for us. And as mentioned, Josh and Justin will also join to say a few words at the end. So as a reminder, AMX0035, sodium phenylbutyrate and taurursodiol, is an investigational drug in EMA and is not approved for use there. This presentation is intended to provide scientific information, and some of the information in this presentation has not been fully evaluated by the FDA or other health authorities. So as we were thinking about an Amylyx update and what that would include, it felt like we should start with the year in review because a lot has happened this year for Amylyx. We started out the year with the phase 3 Phoenix clinical trial already underway. That started last year, right around this time, actually, and kicked off the year with a MAA submission in Europe, and then that being validated for review in February. That is still under review in the EMA, as mentioned. In the middle of March, we were able to begin our US expanded access program. At the end of March, the first advisory committee was held. In the middle of June, we received Health Canada approval for AMX0035 with conditions in Canada. The second advisory committee was in September, FDA approval last week, and here we are at ALS ONE now at the beginning of October. And so what we thought we would do today, with all of this in mind and thinking about what we've learned as we thought through our data and presented it very publicly with the two advisory committees as we've had regulatory discussions, is just pause, reflect, look forward. What have we learned from CENTAUR about our therapy, sodium phenylbutyrate and taurursodiol, and also from the post-hoc exploratory survival analyses that, for those that were tuned into the second advisory committee, were a big part of that presentation and discussion. And also talking through what's next, especially for two key US programs, the phase 3 Phoenix trial for US participants, and then the US expanded access program. And of course, happy to take questions as well too. Before I dive into the data, we thought we would hit on a few nomenclature items. We've got a few names out there, which for me as the head of scientific communications drives me a little crazy, but we will make sure that we explain them and that everyone knows why we've got different names in different places. So really the nomenclature is going to differ based on geographical region. In the United States, you'll see us using sodium phenylbutyrate and taurursodiol. You'll also see us use taurursodiol in any publications as well. That is Relyvrio in the United States. In Canada and in Europe, the name is sodium phenylbutyrate and ursodoxycoltaurine. In Canada, that is known as Albrioza, is the brand name. And this is just sometimes in terms of regulatory agencies, you just end up having two names. There's nothing different in terms of the therapy. And a reminder, PB and TURSO is investigational in EMA, so there's of course no brand name there. The MAA was submitted in January, validated in February, and still under review. So just some quick basics on what have we learned from the CENTAUR trial. So in terms of study design, this was a multi-center trial in adults with ALS in the United States, run with the NEALS Consortium. And primary outcome was the ALS FRSR. A few important things to note about the study design. Key inclusion criteria, as noted on the left-hand side here: definite ALS, 18 months from symptom onset, SVC greater than 60%. This was designed to enroll a homogeneously and fast progressing population of people with ALS. And standard of care use of riluzole and edaravone was permitted during the study. So the results that we see are on top of that standard of care. The study was randomized 2-to-1. So PB and TURSO plus SOC, 89 participants versus 48 in the placebo group. And a 24-week or 6-month study. So, really the main outcome here is the primary endpoint, which is the ALS FRSR, where we see a 25% slowing of disease progression as measured by that total score. Shown here on the slide, we have the graph with PB and TURSO in blue and placebo in gray. And of course, always important as well to assess safety. The safety shown here is reflected in the FDA prescribing information. The most common adverse events, that's at least 15% and at least 5% greater than placebo, were diarrhea, abdominal pain, nausea, and upper respiratory tract infection. So as you can see, mostly GI related. These GI-related adverse events occurred throughout the study, but they were more frequent during the first 3 weeks. And as shown in the table on the right, again, the GI adverse events compared to placebo are the most common. And those are percents in the table, just given the 2-to-1 randomization. Additional safety that is also included in the US prescribing information. First point is that taurursodiol is a bile acid. And so patients with disorders that interfere with bile acid circulation could have a risk for worsening diarrhea when taking PB and TURSO. And so these conditions include enterohepatic circulation disorders, pancreatic disorders, or intestinal disorders. And so really the advice in the prescribing information is to monitor for new or worsening diarrhea in folks with these conditions. PB and TURSO does have a high salt content. And so there is also advice around being aware of patients who are sensitive to high salt intake, those with heart failure, hypertension, renal impairment, for example, and considering the amount of sodium in the dose and monitoring appropriately. So the next thing is over long-term follow-up. And, I think I mentioned really over the first 24 weeks, main outcome was the ALS FRSR. We also, over long-term follow-up, have another, of course, very important outcome, which is survival. And so we've really looked at the survival data from CENTAUR in a number of different ways to fully understand it. And to fully characterize the data. And so I'll kind of go through our thought process there and the results that we have. So starting with week 24, so in CENTAUR, the study was designed such that there were a limited number of deaths that were anticipated in those first 24 weeks. It was actually an inclusion criteria. So we did not anticipate to see a large number of deaths, and that's what we saw. We see 6% in the PB and TURSO group and 4% in the placebo group. And these deaths do appear to be related to ALS disease progression. There was a pre-specified composite endpoint of death, tracheostomy, or hospitalization at week 24. We see perhaps a trend in the right direction there, but not achieving nominal significance, which again, wasn't super surprising given that there just weren't very many events in those first 24 weeks. So then let's look at survival over long term. And this is something Dr. Cudkowicz mentioned earlier, which is, I didn't mention it on the study design slide, but after the first 24 weeks, participants who completed that initial phase of the study were able to continue into an open label extension phase where they were able to receive PB and TURSO. And so we were following participants for a long time, up to 42 months from randomization. And so when we thought about a long-term survival analysis, there's a few interesting things that were done in this study that I think are key learnings to take away. One was being able to assess survival status in essentially every participant. So we were able to use a participant locating service to get vital status, which is if someone is alive or if they have died, as of the analysis cutoff date. So the long-term survival analysis that we did in the ITT population, we've got data for 136 out of 137 participants as of the cutoff date of the last participant last visit. So this is a bit of a simple comparison. We're really looking at, did you start on PB and TURSO or did you start on placebo, and comparing the overall survival between those two groups. This analysis, therefore, is not able to take that crossover in the open label extension phase into account, which I'll get into in a bit. And another important item to note, which is another kind of one of our key learnings, is that investigators and participants were blinded to their original treatment assignment throughout the study. So with this, did you start on PB and TURSO? Did you start on placebo comparison? We do see a longer median overall survival. And when we're looking at the last participant, last visit cutoff date, the difference between the groups is a difference of 4.8 months. So 4.8 month longer survival for that group of participants who started on PB and TURSO. And so you'll see in the prescribing information that it is noted that this is an exploratory analysis that should be interpreted cautiously, giving the limitations of data collected outside of a controlled study, which may be subject to confounding. And so with that in mind, and also with the other limitation of not being able to account for placebo group crossover, really what we've thought about for the better part of the year is, are there other methods that we can use in looking at the survival data to, one, account for placebo group crossover, and two, to support this ITT result that we see. And so the analyses that I'll now present are really approaching those two questions. And to get to the second one, we looked to external controls to provide further support for the ITT result that we saw. So 3 additional post hoc analyses. There's the rank preserving structural failure time model, which is an adjusted placebo arm. So this is not using data new to the trial. This is using the ITT survival analysis model used in oncology, and you're adjusting the placebo arm for crossover. The other 2 analyses are using data external to the trial. So we've got the NCAL survival prediction model, which is giving us a natural history control. And then we have a propensity score matched PROACT historical clinical trial control. And so for both of those, we're able to, sorry, moving this thing out of the way here. For both of those, we're able to, again, have that external comparator to hopefully provide additional support for the ITT overall survival analysis. So for the NCAL's prediction model, what we did there is generated the predicted survival for each participant in CENTAUR using that model as it is publicly available. And then we're comparing the predicted survival for that original 89 people in the PB and TURSO arm to their observed survival in the same arm. For the propensity score matched PROACT arm, we're creating a synthetic control arm from PROACT. We're taking participants from PROACT based on key criteria, and then propensity score matching even further, and comparing the survival of that now PROACT control arm again to the observed PB and TURSO survival. Each of these has strengths and limitations, and that's one of the reasons that we approached in these 3 different ways. Of course, all are post-hoc. The RPS FTM does assume a common treatment effect, which is basically no matter when you start therapy, you would see the same benefit. That, of course, in a neurodegenerative disease could be an assumption that could be questioned. However, given that CENTAUR is a 6-month study, we felt that that was a reasonable assumption. NCALS is, of course, a European clinic-based population, which we're comparing to a US clinical trial population, so there could be some differences there. And then with PROACT, I think one of the key limitations that was discussed is just the time frame of data collection, and if there's been improvements in standard of care, improvements of survival over time, if that would be reflected in the analysis. So looking at the data in terms of the results that we see from these analyses, at the top, we have the ITT overall survival analysis. That's the original one I shared with the 4.8-month difference. And then the next few lines, so the RPS FTM, that's our adjusted placebo arm. When adjusting the placebo arm for that crossover, we're seeing a difference of 9.7 months in survival. With the NCALS natural history comparator arm, we're seeing a difference of 9.9 months in survival. And then with the PROACT propensity score matched arm, we're seeing a difference of 11 months in survival. So one thing that we found interesting was, despite using these pretty different methods and different approaches, we're seeing concordance in the survival difference between these groups. So that sort of gave us a little additional confidence in terms of the methods that we chose, and then also, again, provide that support for the original ITT survival analysis. So, of course, the data is interesting and understanding, the potential survival benefit for PB and TURSO, but I think there's also some important learnings just from the analyses themselves and also from the discussions that we've had about the analyses. And so, of course, one is balancing capturing that survival data while also limiting time on placebo. And so the way that it was done in this study was the short 6-month randomized phase within the open label extension phase, allowing for that longer term survival follow-up. With that, however, we did maintain blinding to the original treatment assignment throughout the end of the OLE phase, which could be an important learning for future studies as well. I know we've talked about this a lot, and I know Dr. Paganoni has presented, the participant locating service, allowing for that comprehensive data capture. I think that's another important point, and perhaps something that other studies can also look to in the future. And then just thinking through these additional methods, the RPS FTM, which is a method very frequently used in oncology, looking at our natural history data sources for comparator arms, and how we can potentially utilize these types of methods, perhaps even in a pre-specified way, to sort of support the survival difference that we see and also to account for placebo crossover. So I'm sure there'll be a lot of additional interest there as well too. And I thought I'd just take real quick a few minutes just to talk about what's next, because we know it's only been a week since FDA approval, and there's some, of course, immediate updates and impact that we wanted to ensure that. I somehow got muted, sorry. So for the Phoenix trial, I think very important to highlight that it's underway. There is no impact to Phoenix in Europe. Recruitment is ongoing. The majority of Phoenix participant recruitment has been in Europe and will continue to be. The Phoenix open label extension will continue in Europe as well too. And no change in terms of when we anticipate top line data. that's still in 2024. Recruitment is closed for Phoenix in the United States. I know our Amylyx team is working hard to be in contact with US trial sites. There have been webinars already this week. And please, if you have any questions, reach out to your site coordinator. If you're a person living with ALS, your physician, and the Amylyx team is definitely here to help assist with questions and with transition in terms of that trial. In terms of the U.S. Expanded Access Program, individuals who are currently participating in that US EAP will work with their clinician to determine if they wish to remain on treatment now that PB and TURSO has been approved in the United States. And again, similar to Phoenix, Amylyx personnel are in contact with sites. We've had calls and webinars, and please continue to reach out to us if with any questions and anything that we can help with there. And with that, I'm sure we left a logo off here, so please accept our sincere thanks for the participants and their families who gave up their time in CENTAUR, to the study sites, physician and staff who helped with that trial, to our ongoing Phoenix trial, TriCals and NEALS and their important role there. And then we would be remiss to not acknowledge Dr. Cudkowicz and Dr. Paganoni. And I'll turn it over to Josh and Justin in case they would like to add any final words there.
Not much else, just to really say thank you. So many people on the call here have been collaborators for a long time, and truly none of this would have been achieved without them. And Josh and I were sort of welcomed into the ALS community many years ago, and we really knew absolutely nothing about clinical trials or clinical development. So much of what we've learned is thanks to the many of the amazing people here, and we're really grateful for that. And we also really hope that this is just the start. We have a lot more work to do. And I also have to say thank you so much to Jen and the team at ALS ONE. This is always one of our favorite conferences of the whole year. I think the way that you all bring the community together, we have great science and companies present, and there's great data and clinical data and discussion. It's just really wonderful. So thank you.
Just echoing Justin, I think, rounding out with the data, the ultimate approval, I think the main emotion, at least Justin and I felt, was gratitude. There's been so many people who have mentored us, guided us, taken time to, help this drug and this company along. And, I think as we, continue forward, our goal is to continue in close partnership with everyone, to continue learning and listening to everybody. Please always give us any feedback, you feel is appropriate and otherwise. We hope this is just the start for us. We're thrilled about this drug, but there's still a lot to do in ALS, and we want to keep working hard and fighting hard until this disease is no more. So thank you all so much, and I think we have questions.
Yeah, so well, thank you all three of you. Jamie, thanks for a wonderful presentation. We have a couple of questions. I'm going to take just a minute because there are a few questions that we hear sort of a lot here. One is kind of, I think, as the broader medical community hears about this drug, they've been asking questions about the fact that it is sodium phenylbutyrate that's in it, and there is a salt load that comes with that, and I wondered whether there was any signal in the trial that you could point to that would say, that we saw anything related to that salt load of it.
Sure. Yeah, I'm happy to take that one. And Sabrina, welcome if your thoughts there as well. But my experience with looking at the data is that we did not see any indication that the amount of salt was causing an imbalance in terms of adverse events or discontinuations. really the main signal that we see are the GI adverse events. But with that said, in a real world population that is not as controlled as a clinical trial, it's certainly important just to be aware of the salt that's in the product.
And if I can just add one thing to
Yeah,
that, absolutely, I agree. And just to put things in perspective, the full dose is about 900 milligrams of sodium. And for people who have, let's say, uncontrolled heart failure, the max daily dose, depending on the guideline that you look at, is around between 2,000 to 3,000. So again, certainly the amount of sodium in sodium phenylbutyrate is something that's worth mentioning for real world application of the drug. That said, it's well below the daily limits, even for people who are on a salt restricted diet. And obviously that can be compensated by diet as well.
Yeah, I think that's a really important point too. There was one question about taste and whether there, what the future is for taste. Are there any options for taste in the future?
Yeah, that's certainly an area of interest. What we can say is, of course, at the moment we do have taste mitigating strategies that worked with the product that's approved, and so we're using the same ones that were allowed in the clinical trial in terms of when we set expectations with people who will be taking Relyvrio, and also with physicians that will be prescribing. So there are some ways to mitigate what we're currently using, and certainly an area of interest for Amylyx as well too, to evaluate potential new formulations down the line. Justin, I saw you unmute. I don't know if you want to add anything.
Yeah, you did. So thank you. I'd say too. One is, I think in both our the ALS studies, but also Alzheimer's, there's still pretty good compliance, and I think Dr. Paganoni, Dr. Perry, others could talk about that better than I could. But so I think it can be managed, but it's definitely real, and so I think it's important to address. And it's why, as Jamie said, it's high on our minds of how can we make this better. And so we're already actively working on a new formulation that would be considerably more palatable, and everything that's regulated takes much longer than one would hope, but our teams are working really hard on that because we don't want people to have that as a challenge.
I guess I would just add that as an observer, there's haven't spent a lot of time studying or thinking about taste, but there's been a wide variety of how people have reacted to it. It's taste is not a monolithic thing, is what I've learned from watching people's experience with the drug. But good to know that it's high on mind and something that you're working on as well. I think we have to move on to there are more questions. Actually, there are questions in the chat. Maybe we may be able to get to those later or offline. But I want to move on. So Sabrina Paganoni, who once again doesn't